Welcome to Ask the Expert. It's a really brief, informative, lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording the event. We're going to post it later on our YouTube channel, so you can watch it there if you missed some of it or all of it. And if you have any questions for our guests, feel free to enter them in the chat, raise your hand at the end of the presentation. And today we have as our guest, Dr. Ester Caffarel-Salvador. She's a multidisciplinary scientist with a background in biotech and biochemistry. She was actually recognized by the MIT Technology Review with 35 under 35 innovators in 2019, which is a huge accolade, and the Nova Talent Award in 2021. She was postdoc in Robert Langer's lab at MIT, and she developed a novel pill designed to administer insulin and other macromolecules via oral delivery in collaborations with Novo Nordisk. She's now an associate director at regenerative medicine at Leo Pharma, and she's really passionate about advocating on career development for women in science and is an advisor and mentor to various startups and academic programs. So wow, you've done a lot in a very short time. Thanks for joining us. I didn't know. Yeah, sure. I didn't know if you wanted to give us a thumbnail sketch of any more of your sort of career path and how you got here, how you got to where you are right now. And then we'll chat.
Sure, definitely. Well, I did my undergrad training in Barcelona, so biotechnology and biochemistry there with a year in Italy. So then is when I hopped to the UK, where I ended up doing my PhD, and it's where I first got introduced to microneedles. And at the beginning, I was like, "What's that?" That at the time the field was much more smaller, more like a niche, and now it has bloomed with so many technologies leveraging this approach. And actually, this expertise in microneedles brought me to the US, MIT Bob's Langer Lab, where I was involved in a project to deliver insulin orally, so designing novel devices that enable to intake insulin orally as an alternative to injections. And that's the work that I will be talking about today. But then like the expertise in drug delivery across skin and orally, but also like led me or set my path towards going back to dermatology. And now I'm just researching in the field of regenerative medicine for dermatological diseases. So I think there are many opportunities of improving the life of patients. And now I focus back on dermatology, but still very passionate about oral delivery and devices and helping out the patients.
Yeah, I mean, I just spoke to a group that was very interesting, John Glass and Yo Suzuki, last week, and they were talking about transdermal, kind of applications that they're lotions that they're sort of developing. So there's a lot of avenues that people are exploring. Yeah. So do you want to give us share some slides with us?
Yeah, absolutely. Let me do that. Okay. Can you see my screen?
Yes.
Sure. Perfect.
I do. Okay. You might want to just put it on full screen though, because I can see like two sides.
Wait a second, because I have two screens. I think it went to the wrong screen.
No worries. Okay. This is always a little bit of a transition.
Okay.
Perfect. There it is.
Great. There you go. So yeah, today I wanted to briefly touch on oral insulin and the novel devices that I've been working on. But for that, I wanted to give a bit of context first. So efforts have been made for almost a century to address the problem of delivering biologics like insulin orally. And already in 1923, a scientist named Harrison tried administering insulin orally in an alcohol solution. However, he had success in only one of the 4 patients. So consistency was an issue there. And since scientists have been trying to look for alternatives to injections. So when compared to injectable drugs, oral formulations acting in the same biologic pathway and for the same indications are more commonly prescribed. This is so even when the injectable drugs demonstrate clinical superiority results. For example, in the treatment of type 2 diabetes, in this case, what I'm showing in the slide, is that here you can see the GLP-1 receptor antagonist that is an injectable, and those pills are the DPP-4 and DPP-4 inhibitors, and both are to treat diabetes. But prescriptions are higher for the oral route, but this entails that efficacy is being compromised because the injectables work better. So there is a need to overcome these barriers, and we shouldn't need to compromise between choosing on efficacy versus administration route. So bearing that in mind, what I'll be talking to you today is about 3 devices that we worked on at MIT, and will be a brief overview to give you an idea of the constraints that we are trying to address for the oral delivery, and also hopefully have a discussion around this. So I'll be presenting about these 3 devices. So the first one is a device for delivery in the buccal space. The second one will go to the stomach, and the third one into the small intestine.
Can I ask you briefly, like, what is the scale of those?
I have actually pictures that show it, like, to, you know, in comparison to penny later. So I think it will be easier to see. But
great.
all
Yeah,
these
let's go.
fit in a capsule, so it's just very small. Even if here they seem bigger, like they would all be swallowed in a capsule that are being currently used to deliver other medications.
Okay, cool.
So just in order to understand like what's needed to design these devices, I want to highlight that the gastrointestinal tract, or the GI tract, is designed to break down food. It is inhospitable for most of what it goes through it, and therefore, like insulin as a protein and like many other components of food, it's aimed to be broken down by all its enzymes and the different pHs that we find in the GI tract. So the GI tract is composed by several organs that go from the mouth through the rectum, and those are the mouth, the esophagus, stomach, small intestine, and colon. And in this table, I just aim to show a bit the diversity of like the different tissues, because it's very important in order to design a successful device for drug delivery to understand where it needs to go and what are the properties of that tissue. You can see that the thickness of the tissues varies across the different organs, the residence times as well, like it's just a few seconds that we have a pill in the mouth or the esophagus, while it can stay between 1 and 3 hours in the stomach, and then the time to transit through the guts is much longer. Therefore, there would be a delay if the aim was to target such organs. Absorption also varies across these organs for the pH, as we will know the pH in the stomach is very acid from this degradation, and that's why many of the drugs cannot be delivered orally, because they never get absorbed like when they are active to carry out their function. Similarly, a lot of enzymes in all the organs, and there are mechanical forces, right, associated with either the swallowing or like the pumping of the stomach. So with this, like, let's take a closer look at some of the devices. So the first one is based on buccal microneedles. And the concept, what we aim to deliver here was a buccal patch. And we do this with the microneedle patch that looks like this at the skin. Here you have a real one, and actually this is 1 centimeter per 1 centimeter. And these microneedles that here they seem very big are just tiny, like about 1 millimeter in height. So you can think of these like a Velcro patch, like on your clothes that you apply usually in the arm, but like we've adapted these systems to apply them also into the GI tract. So the idea of this patch is that these tiny needles have the drug, insulin in this case, in the tip. And once you put them against the mucosal of the mouth, they dissolve, and then they deliver the insulin that then gets into systemic circulation.
How does it feel?
You barely feel anything. Actually, microneedles are short enough that they don't reach any nerve endings, so you don't feel any pain, and there is no bleeding associated with them. So many people describe them as you know, the aspros, like the tongue of a cat, or like if you were rubbing like Velcro against your skin. So you know, you feel something, but it's not a painful sensation.
Yeah, probably a lot less than an injection.
Yeah, that's right. So yeah, in this case, we targeted the mouth because it was easier to penetrate that skin. That is where usually microneedles are being applied. The buccal mucosa doesn't have the stratum corneum that makes the skin so elastic and more difficult to penetrate. Also, the dissolution rate in the mouth is much faster because it's moist, and it goes, well, it's very accessible, and it goes straight to be delivered into systemic circulation upon absorption. So this is just the patch that we loaded with microneedles, and I just don't want to go into details of the data, but what we did was first validate this in pigs. We use pigs as a model drug, and also insulin is very useful as a model drug, but we can think that these patches could also be used to deliver human growth hormone, for example, for the pediatric population as an alternative to injections. And what it enables us is to monitor like the blood glucose drop, instantaneously post-delivery, while to actually prove that it works, as we can see here, like these samples get processed sometimes like weeks or even months after they are collected because they need specific assays. But basically just this slide to show that the peak with the microneedles was about after 40 minutes approximately, and it gets a delivery similar to the subcutaneous injection. But the exciting also part about this specific project is that we got to test these patches in humans. And I wanted to show you this slide to explain how we did that, because we had to design a system, an applicator, to ensure that we would be able to test it in a reproducible manner to each of the volunteers. So we recruited 100 volunteers at MIT, and this was the applicator, and it had a light coding that would enable us to know that all the microneedles were applied with the same force and with a specific time frame. And we designed these in a kind of toothbrush shape because we knew it was accessible in the different parts of the mouth. And what we did is we delivered a patch without microneedles and a patch with microneedles, as you can see here. And we wanted to see if like the volunteers feel the difference between them. Like we wanted to see what was the discomfort perception of both patches, but they didn't know if we were applying them with microneedles or without them. And we also tested different locations within the mouth. So these are the locations that we tested, and I'll go through this graph in a second, but like basically the palate, which is the top of the mouth, the cheek, the lip, tongue, and lingual. And you might be familiar with these Lego graphics here that you can see at the PCP office. And it's mostly to assess what's the discomfort associated with a specific injury, right? And so we explained how these work, and we asked also our volunteers to draw a vertical line on how they felt about that specific patch. And here is the results. We quantify these from 1 to 10. And you can see that like the preference, like we compared it also to skin. We use skin to explain them what was going to happen in their mouth. And the palate was the preferred application side, followed close by cheek. And the values didn't differ that much, especially in cheek and palate when we used the patch with microneedles and the patch without microneedles. So that was very exciting to see. And also like the volunteers reported 95% of them that they would prefer the microneedle patch over the injections. I need to highlight that 60% of the participants didn't mind needles, so they would still go and get a flu shot, or that they were comfortable receiving an injection. But many others were just wondering when would that be available to the market to get a flu shot or any of the other injections that they need to acquire.
Yeah. And I mean, in terms of diabetes, it's a chronic, injections, not just like once a year getting your flu shot. It's just, it can be constant, replacing sites and, giving yourself injections. So this is great news. Really nice.
Absolutely. Like that would be very accessible, easy to deliver the patch within the mouth, and like the GI tissue regenerates pretty quickly as well. So that would be a good alternative. So hopefully these devices also will just make it at a fast pace to the market and manufacturing, and pose an alternative to these uncomfortable injections.
Yeah.
And then just the flavor as well of this device, which we name Soma, which stands for self-orienting millimeter scale applicator. And as you can see, and this seems like pretty big, but it's one of the conventional capsules that usually they are painted white and blue, or like that they tend to have powder inside. But these would be able to embed a couple of our Soma devices for delivery in case like more dosage was needed. But like here, we wanted to choose like that, you know, those are very small and you would barely notice them. And they wouldn't be transparent, the capsules, obviously. Everything would be more like prepared towards the patient and just optimized. So this was one of the first prototypes. But here I'm just showing a diagram of how does this device work. And I need to say that it was inspired on the leopard tortoise that you see here, because these turtles have the ability to self-right themselves. And this is what this device does, as I will show you in a second. But the reason is because like the center of mass is pretty low here. So it's made of a biodegradable polymer, all these gray shell. And then in the bottom, it has a stainless steel base. So this means that it weighs more and the center of mass is down here. So it doesn't matter what position gets delivered into the stomach, that it would always make sure that it's in this upright position and this part will be against the stomach wall. So what happens inside here is that we have a spring that is embedded in sugar. And here there are these vents that enable the liquid from the stomach to enter. So once this liquid enters, dissolves the sugar and the spring deploys. And then what happens is that this needle elongates, delivering the insulin, which would be the light green part. So this blue part is just support. And the insulin payload in the solid state would be here. So it doesn't need to be a liquid like we have pure insulin that would be like a depot inserted into the stomach wall. And like this, there would be absorption. It has no time to be exposed, you know, to the pH, the low pH of the stomach that we were talking about, or all its enzymes, because it gets delivered into the wall where it goes straight into systemic circulation when it dissolves.
So this time frame of, oh, here, you're getting to it. Exactly.
Yeah.
Yeah. And also, you know, how much, what is the payload that these little, you know, capsules can carry?
So we can load more than 1 milligram, like, and we have optimized this version. Also, there is a liquid version now. So the payload is even further increased, but like this dose is like not with, like it's far above what an injection of insulin would be needed. So, you know, but also it could be adapted to be long-lasting insulin, maybe the shot in the morning, you know, the basal insulin that dissolves over the course of the day. So you can tune a lot the properties of the activation rather than feeling like a single shot. So it would be more like, you know, yeah, for example, the first pill that you take in the morning as your basal insulin for the day, like you could take it in the form of a pill. And what you see here is like what would happen like with this device when it lands into the stomach. So as I said, the center of mass is in the lower part, so it doesn't matter which position it falls from, it would always revert to go to the stable position. So basically this stable position is reached within 1 second. It's like really fast. And then within 1 minute, the injection occurs because that embeds the spring is dissolved within 1 minute, and then the injection goes inside the tissue, and then the insulin can dissolve approximately within an hour. And this is tunable, as we said. So here we were just validating the proof of concept of our Soma device.
Yeah.
And similar to what I showed before, like we saw a similar delivery as an intragastric injection and a correspondent glucose drop.
And then did this get into clinical trials with humans?
So not yet, like the process is much slower. So like this was validated in swine again. We have pigs that we validated with, and like we also dose the pigs with several devices at the same time, 6 or so, when we, you know, make sure that these went through all their body, that they were excreted, didn't cause any collapse into the GIs. We looked into this closely. We also analyzed all the gastrointestinal tract to make sure that there was no bleeding or tissue disrupted from this injection. So there was a lot of validation, but usually it goes into a second animal model before making it to clinical trials. And like the first step into clinical trials will also be ingesting such devices with no drug to validate that they are safe as well. And then on the second stage, the drug will be added as well. And this can serve as a platform. So we validated also with insulin due to our collaborator, but you could think that, also potentially vaccines or other therapies might be embedded in there. So they could also revolutionize how many drugs are being taken.
Yeah.
But hopefully they will make it to clinical trials soon. That would be great to see in the upcoming years. But Novo Nordisk, which is our partner with MIT, are looking to progressing this technology to the next step. So stay tuned for that, I would say, and hopefully we'll see these advances.
Yeah, no, these are really interesting and creative approaches for sure.
Yeah, so we just design different devices, because at the end of the day, there are different drugs that you could accommodate this from, and ones might be needed for a faster delivery, and others, for a more targeted delivery or based on absorption. So just to finish giving you a flavor, I'll be talking about what we call LUMI, and this stands for luminal unfolding microneedle injectors. Just think of these microneedles that I showed you for the buccal space as well, these tiny projections. We've designed a device with microneedles at the tip of the arms, and no worries, I'll zoom in into the next slide for that, but just so that you understand how this works, it's inserted in a capsule. It's an enteric coated capsule, and what this means is that it goes past the stomach without opening. Basically, the low pH doesn't affect the capsule, but when it goes into the small intestine and this pH raises again close to 7, the capsule dissolves, and it's when it delivers the device. So the device has some microneedles at the end of the arms, as I'll show, and once in the small intestine. These also are delivered to the intestinal wall. The drug is dissolved, and then the rest of the arms are also dissolved, to enable the small pieces to pass through if needed. So here is where you see the device, and I'll show you a real picture compared to a real scale object in the next slide, but so that you get an idea, this is based on a star-shaped device that has an elastomeric core. So basically, this is closed inside the capsule. So these 3 arms all in one, so they all fill in a similar capsule to the one that I've showed you for the previous device. And once this capsule is dissolved, these arms open. And these have been thoroughly thought. You can see that the edges here are tilted to accommodate these needles. And this is because, during the release of the device from the capsule, these needles are not shredded with the capsule, and that's why there is this indentation, and also that they are targeted towards the small intestinal wall. And these arms are also dissolvable, so before, the insulin or the drug would be towards the tip of these needles, and there is no drug in these arms, but also those are made of polymer, so they dissolve and disassemble once they've release the cargo.
This is really interesting because at the onset of type 1 diabetes, there's a sharp decrease in the diversity of the microbiome. And there's been a lot of talk about repopulating the bacteria that actually are killed off or whatever happens to them, right? Because they disappear. Right. Can you imagine that this kind of vehicle could deliver those bacterial strains that disappeared at onset of type 1, or is it really focused on delivery of insulin and molecular medicines?
Yeah, that's a great point, a great question. And I think this device opens a wide range of possibilities for these bacteria or drugs that struggle to be absorbed into systemic circulation. In terms of bacteria, I believe that you could just deliver them with enteric coated capsules to repopulate the small intestine, but they wouldn't necessarily need to be localized in the intestinal wall for absorption, right? Because what you want is to populate the bacteria into the small intestine per se. While this device devices aim to, circumvent the fact that if we were to deliver insulin orally, it would degrade. Like, right, if it was just on a pill, powder, or even a liquid, it would just be broken down by the enzyme. So I think that like just a conventional pill that with enteric coating that targets the small intestine might solve the bacterial problem. But like absolutely, like all these devices can be used as platforms. So you could deliver many more drugs than insulin.
It's almost like this delivery setup could sort of be used to like get down to the small intestine and maybe anchor there and then release the bacteria that have been depleted, just for, anchored there for a little while and then release. But yeah, because you don't want the delivery actually into the enterocytic wall. You want it just to kind of be right at the top. So it is really it's a really interesting, way of delivery.
Mm-hmm. Yeah, absolutely. And there are other companies also working on some other devices. I mean, another company is investigating an adhesion like device that would adhere to actually the intestinal wall. So maybe that one would be better suited for bacteria delivery. So we've at Leo Pharma, actually, we've just written a review that will be coming up in the next couple of weeks that also compiles all the oral drug delivery devices under investigation, and like they target from the mouth all the way through the colon, and each one sort of there are variations. Some are just based on liquid formulation delivery, others like more on the solid state, or just designed to stay slightly longer into the small intestine, for example. So I feel like this space that also it's gaining popularity and interest on exploring, finding alternatives on the delivery of many macromolecules. So
there's a lot of room to explore and invent, really, it sounds like.
Exactly. Yeah, no, and these are very novel. It was like we started kind of with a whiteboard and started like populating it with ideas. And obviously there were a lot of failed experiments and a lot of trial and error to make sure, that like this device, for example, is deployed, each time that it's deployed, like gets the microneedles inside the small intestine. So it was, very important to make sure that, for example, the elastomer core had enough strength to pop this open and make sure that they made contact. So, we did a lot of tests like that, that the arms wouldn't dissolve prior to delivering the microneedles that at the edges. So, there is a lot of development. I'm just showing, an overview and obviously showing insulin delivery, but the engineering work and the validation models and everything that was behind it, are much more extended. And there are like, some modifications of these and other devices from other research companies that are also like very exciting to see.
Yeah, for sure.
So, going back to this device here, we can see the size of the Lumi capsule as well. I mean, it's similar to the dimensions that I showed previously, like, and comparable to the time here. So once it opens, it deploys and it extends. But, we, in order to design how long this would be, would take into account, the measures of the small intestine and make sure that, it won't fully extend like its open position, but just enough so that the needles, the microneedles that you see here, that again are less than 1 millimeter, they will be inserted into the small intestine. And one thing I want to highlight is that the GI tract doesn't have pain receptors; it has stretchability receptors. You wouldn't feel these needles whatsoever, even the one from the Soma being delivered, because there are no pain receptors. So that's something like in favor of taking in those devices.
Yeah, it's an advantage.
And basically, obviously, like to prove that we deliver insulin here, I'm just showing some of the data. Here we start with the blood glucose change. So we can see that there is a decrease with the Lumi. What we compared was with the device, so the one that there is no decrease here delivery is just because what we did was dissolve the microneedles that we would usually mount into the Lumi device into a solution and then deliver that solution. So as if it was, orally delivered, taken in a liquid insulin, and we can see that there is no blood glucose drop or no insulin absorption when we deliver insulin in an aqueous solution. While when we do it with our Lumi device or or a patch inserted into the small intestine, we can just see a peak of insulin, and it's comparable to the sub-Q injection that you can see here in the dotted line.
Yeah, that's a great profile.
So yeah, in summary, insulin is a macromolecule and very difficult to deliver without the use of hypodermic needles. Nonetheless, science is progressing fast, and even if we have not discussed today permeation enhancers, permeation enhancers also offer a possibility of, increasing absorption to the GI tract. However, oral devices go a step further because they enable targeted delivery and increased absorption, resulting in higher drug bioavailability. And here are the three devices that we briefly reviewed. And for further information, you can refer to the papers like under the pictures. And as I mentioned, there will be another review coming up, this time from Leo Pharma, that we actually compile like the several devices that enable oral delivery of novel molecules. So yeah, with that, I thank you for your attention. I'm looking forward to further discussion.
Yeah, no, this is fantastic. I think, I guess some people are wondering, what's next? So what's next for you with these devices? Are you going to bring them into the space of Leo Pharma, or, can you even talk about that?
Yeah, so well, these devices stay with MIT and Novo Nordisk because then they came out of this collaboration that I work on during my time at MIT. So like the new set of scientists or like the company is looking to progressing them, but I would envision that like many other companies might want to leverage these technologies to deliver their drugs per se. So at Leo Pharma, like we actually compiled this review because we are also looking into oral delivery and regenerative medicine. And I mean, if you look at the novel technologies out there, like with all gene therapy and so on, as of now, it needs to be delivered by injections as well. So, if these devices continue to grow up and get validated for these technologies as well, it could be an option. I mean, obviously, we're looking of a further future and there is a long way to go, but one of the other companies that has a device for also oral delivery has it already on phase 1. They've already tested it with patients and make sure that there are no safety issues associated with that. So the next step would be like moving into phase 2 and like continue testing and validating this technology. So hopefully, the devices that I've spoke about today, like will follow the same path.
Can we give a shout out to that company, or should we just?
Oh yeah, Rani Therapeutics.
Rani, and are they in Boston also?
So no, they are on the West Coast, but they have like some partnership. They partner at a few years back with Novartis as well. So I think there are many companies looking into that. Like we have also like BioGrail in Europe that's looking into that by Wind that is also in the Midwest. So a lot of US companies focusing on oral drug.
How can you imagine, here's a question, how can you imagine using some of these technologies to interface with eyelid implantation? Or can you? Because that's one of the biggest problems, for eyelid implantation. Vertex is trying to bring that to market now, is fibrosis and the, the resuming immune attack once the implant is in place.
Right. I feel like in some of the cases when you are delivering a depot, if you might be able, it depends on the cargo and obviously needs to be optimized, for each of the drugs, because when we validate different drugs have different molecular weight, like, stability properties. So you need to make sure that it's compatible with, the device per se, the polymer that is holding it and so on. So I think that in many cases, it will need to be tailored, or maybe it just needs a different device to ensure like that, for eyelid delivery and so on. So I think like at the beginning,
those are like whole cells, right? Versus just a molecule. So it's
Absolutely.
more complex, of course.
Exactly. You need to make sure that the cells are, all stable in this matrix and so on. And like actually in the Langer lab as well, there were other, scientists focusing in like islet cells as well. So there is a lot of research on diabetes from different angles, because I don't think that one solution necessarily is above each other, but like it's interesting to like keep these different avenues of research open and see what just solves the problem better, or like, from different people, like different solutions might be needed than one based on.
Yeah, the multidisciplinary approach. And, we try to foster that here, by discussing all these different options and avenues with, the BME people, the GWAS people, the cell biologists, physiologists. And so, we're attempting to create a nexus here, a place, where all different voices can be heard. And then, different scientists from different disciplines can kind of like tune into that and think, how does this impact what I'm doing? So it was just a great pleasure to speak with you today, Esther, and thank you so much for giving us your time. We wish you all the best success, and I can't wait to see what you do next.